All right, good afternoon and welcome everyone. My name is Emily Bell and I'm the Communications Coordinator for the Florida Wildflower Foundation. Thanks so much for joining us today for our webinar, Using Keys to Florida Plants. We would love for you to say hi in the chat and let us know where you're joining us from today. For those of you not familiar with our organization, our mission is to protect, connect, and expand native wildflower habitats through education, research, planting, and conservation programs. Our work is made possible primarily through the sales and renewals of the Florida State Wildflower License Plate. And this month, actually, we are celebrating 25 years of that plate being on the road. So we're very excited about that. These funds, along with donations and memberships, allow us to support and create projects that build awareness and knowledge of native wildflowers and plants throughout Florida. We'd like to encourage those of you who find our programs valuable to consider becoming a member, making a donation, or purchasing the State Wildflower License Plate. And if you purchase or have the Wildflower Plate, you're eligible for a membership. So just let us know if you have the tag and we can get you set up in our membership database. Be sure also to check out our website for resources on planting and growing wildflowers, to learn where to see wildflowers in bloom, for upcoming events, and more. We're also on social media, and you can find us on most platforms at FLA Wildflowers. Today is a bit bittersweet because it's our last in our 6-part series, Botany and Context, with Dr. Folsom, but we do have some great webinars lined up. Our next webinar on July 16th will be future direction of the Million Orchid Project, Potential for New Natural Experiments, presented by Director of Research at Fairchild Tropical Botanic Garden, Dr. Jason Downing. The Million Orchid Project is the world's largest educational outreach program dedicated to orchid conservation. Also, stay tuned for many great field trips coming up. Our next field trip will be this Friday, June 20th at Alligator Lake Recreation Area in Columbia County. And you can celebrate the summer solstice with me as we're looking for sunny blooms of the American lotus. Registration does close today at 4:30, but there's still some spots available, so sign up now if you want to join us. Also, save the date for a refreshing adventure, wading through Deep Creek in Putnam County on July 19th. We'll likely see several listed plant species in this rare Central Florida Atlantic white cedar hydrant hammock. Also, I just wanted to note real quick, if you're in the Gainesville area, you can find us this Saturday, June 21st at Pollinator Palooza at the First Magnitude Brewing Company. We're partnering with the Florida Museum for a native plant giveaway. There's also going to be a special pollinator themed beer release, live music, food trucks, and other vendors. So we'd love to see you there. Be sure to follow us on social media, subscribe to our newsletter, or check out flawildflowers.org/events to stay abreast of our webinar and field trip opportunities. Just a few housekeeping items before we get started. All attendees are muted with cameras off. If you have questions, please use the Q&A feature to submit them. The chat is active, but we don't monitor it for questions, so you've got to use that Q&A. We'll address questions at the end of the talk if time permits, but you can also email questions to info@flawildflowers.org if your question's not answered. This webinar is being recorded. It will be available on our website and YouTube channel in 24 to 48 hours. When it's available, you'll receive an email from us with a link to the recording, along with a resource page and links from the webinar. Now, without further ado, but right before I introduce Dr. Folsom, I did want to share that we have set up a field trip with a plant excursion with Jim on Saturday, October 25th at St. George Island State Park. We will likely open this for registration in late August or early September, but we did want to note that our members will get a week's advance notice to register. So if you want to make sure you get that early notification, join now so that by August, you'll be in our database and you'll get that early notification. So at this point, I think Dr. Folsom needs no introduction, but we're going to do it anyway because his history is so fascinating. He has a lifelong love of plants reflected in a BS in botany from Auburn University, an MA in biology from Vanderbilt University, and a PhD in research botany from the University of Texas at Austin. Though his research has centered largely on the orchid family, with much of that time spent in tropical America, including a year in Colombia on a Fulbright predoctoral fellowship, Jim's botanical interests are wide-ranging. For 35 years before his retirement in 2021 as curator/director of the Botanical Gardens at the Huntington in San Marino, California, he dedicated much of his effort to educational programs that increase public interest and understanding of the science, culture, and history of plants and gardens. Topics reflected in his website, botanyandcontext.com, as well as his TikTok channel at Botany and Context. He lives on St. George Island with his wife, Deborah, also a botanist, and they have 2 adult children, Molly and James. Jim currently serves as an incoming member of the board of the American Horticultural Society. So without further ado, I will hand it over to Jim.
Okay, let me see. Let me find I got to find the share thing. I've lost it. Uh-oh. Wait, there it is. It appears and disappears magically. Okay. And we have to go back, escape. Let me go back to the beginning here. Sorry. Okay. Try again. Are we there? Can you see that title slide?
We got it.
Okay. So this is the last one, and it's been nice putting together this information. I have learned a lot, and it's really got me up to date in many regards in as to what's happening in native plants in Florida, the research and activities. So that's been wonderful for me. I hope you've enjoyed some of this. And if you, I know it's been a lot of information, but it's all posted so you can go back and check it out, or you can send questions to Emily and Rose, and they can send them to me, and I'll try to make things a little bit clearer if that's needed. Uh, Usually I teach in person, and I really like to teach in small groups in a lab setting where I have available a lot of material and where I can learn a lot about what the students are interested in, because really it's all about you. It's not about me, even though I've enjoyed doing this. It's about you and your interest and what you need or want to learn, because what you want to learn is what you'll learn the easiest and what'll stick with you the best. So I don't know how you got interested in wildflowers or how you came to this program. I'm just made up these things to say that I'm guessing it may have been through friends and family, or you may have been charged up by visiting parks and gardens, or maybe you ran into some printed matter, or you went to a program of one of these programs with the Wildflower Foundation or with the native plant groups, or you just ran into something in social media. So somehow or other you got here and you found us. And so I want you to, I'd like to know what you're interested in as we move along. As I can answer questions on the web, and I can also increase the amount of information that I post on my website based on what people might likely want to know about. So one of the things that's curious to me always is how people learn what they do learn. How do they get the information? What are the steps? For example, are you did you start out just learning how to ID plants or natural history? Are you more interested in biology and ecology? Or you're a gardener? Do you cultivate plants? Are you interested in conservation? Is it all about community for you? How have you learned the names of the plants and the stories of the plants that you know? Because it's the stories that help you connect information and maintain your interest. Now, like I, to me, there's a huge social component. Without these wonderful people out there giving tours and offering programs through various organizations, there'd be you wouldn't have the opportunities you have. Here we have our native plant group. This is the Sarracenia group, and there's David. David has been the spark plug for this group for the last few years, putting together field trips and making sure that people find information and opportunities available. And there's Annie, and of course, we recently lost Lauren Anderson, and he for years was really a mainstay and a great support person for all the wildflower people. Um, there's a lot of information out there. Palmetto's a fabulous source. There's this wonderful brochure that FWF put together years ago, and it kind of people open it and they see not just something about wildflowers in general, but they can actually use it for identification. Many of you might have one of these aids, particularly, I mean, I think it's really common for people to have Hammer's Complete Guide to Florida Wildflowers. That's over 600 different wildflowers photographed. There's also the Taylor Guide, many other books, but those typically go by something like ecological habitat or color. That works for me in certain situations. But I'm guessing a lot more people find the color easier as a beginning point for identifying plants. So if you've done that, you know all about how you use those. You may have gotten into slightly more technical literature. I don't know what sits on your bedstand or hangs out in your car. Um, the Wonderland Henson Guide from 2011, that's the third edition. You may have run into this several volume set, The Flora of Florida, which has got it's the last volume came out in the last 2 or 3 years. You may have this older book, which is fabulous if you live in the Panhandle, and that's the Cluel Guide to the Plants of the Panhandle. Any of these are going to get you in a certain place. You may have ventured online. If you are interested in doing things online, there's iNaturalist. You can purchase yearly a subscription to a guide more geared toward ornamental plants. This is Picture This. Both of these have their merits, and this one, iNaturalist, works better for native plants here in Florida. Picture This is really great when you're traveling around the world and you see something in a garden or in a public landscape and you just don't know what it is. Picture This usually identifies that correctly. It's not so good with the native plants. You may have discovered, most of you probably discovered the Plant Atlas. This is a remarkable program that began in the 90s that, and it's been put together. It's such a good program that several other states have adopted it. I see that Alabama has taken it on and New York, and they've started fleshing out what's in their versions of the Atlas. But the Atlas is amazing. The amount of work that's gone into building that and inputting all the herbarium data and things is remarkable. So that's a great resource. And most recently, in February, the people in North Carolina, Weekly's team, created an app. They have a FloraQuest app, okay? And it addresses different regions. And in February this year, they introduced the one for Florida, and it's 1995. It does some incredibly interesting things, and I can tell you up front that if you buy it, you might want to use it on an iPad or a tablet more than your phone because the tablets give you more real estate. It has keys, and when you find something in a key, it will take you to a photograph. So that's pretty fabulous. Well, that gets us to the topic of keys, and that's what this whole presentation is about. How do you use keys? How do you construct keys? What's the value of keys? How do you reduce the intimidation or the learning curve or the inertial barrier to using keys? We're going to look at several keys and just talk about some aspects of keys. So here is a key from the guide. the Wonderland Henson guide, to Asclepius. So you can see what it is. This is the key. There's the key. The first couplet, it's called. So a couplet is a paired set of choices, and each couplet has two leads. So the first lead is leaves alternate. The second lead, the paired lead, is leaves opposite or sub-opposite or world. Okay. That's not new. That's the way people have thought about Asclepius since we've figured out there was an Asclepius. Here's Linnaeus, 1753, the first edition of Species Plantarum, and his treatment for Asclepius. And you notice leaves opposite, flat. And these are leaves that are somewhat twisted. And over here is leaves that are alternate. So the idea of a key has been around for a long time through the functioning as an outline. Okay. So keys kind of evolved from outlines, but and we looked at one earlier. In one of the previous lectures, we looked at Jusso's way of putting together all the plants in the world and all the flowering plants, and how he sort of outlined those. And basically, that's a key. The first thing that was put together that really is an example of a modern key or a normal key never was published. And that was in 1689. Richard Waller invested a lot of effort into painting pictures of all the wildflowers of Britain and putting together a way to key them based on choices. And he tried to get the Royal Society to publish it, and they never would. So the first key that was published formally and really functioned as a key to plants is recognized as Lamarck's Flora of France, 1778. And he called it his analytical method. And this is huge. The whole book it was 3 volumes, hundreds of pages, and he has reduced all of the plants in the flora at that time of France. And so here's Asclepius. And there were only 2 Asclepius in his flora. One had white flowers, and the other had dark flowers. Curiously or conveniently, this one's called Asclepius alba, and this one's called Asclepius nigra. So that's considered the first real botanical key ever published. Well, that is the beginning of a huge, important program for botanists, because if you're going to study a group and you're going to publish some floristic accounting or a monograph of that group, it is absolutely mandatory in the modern world that you create a key to that group. So here's a group today. Here's a key. Back to the guide. Let's look at polygala. And what I'm doing is I've discovered in my own life that if I'm really interested in a group, a way to learn about the group is to study the key, read the whole key, analyze it, and get a sense of how some other botanist has organized this group and how that person thinks you can identify the plants in this group. So here's polygala. This key breaks it out based on whether the perianth, the flowers, are orange or yellow versus whether they're pink, purple, or greenish white. Okay. And then within the first category, are the flowers in these compound inflorescences, or are they just in these little tight solitary racemes? And you'll recognize these names. Now, what I do, if I'm really interested in a group, and since I'm retired and have time to do things that I never would have done in a previous life, I take the key and analyze it very specifically. But let's look at how keys are constructed. So this is a book that every botanist, every student in systematics back in the previous century would have had. It was a great big thick paperback thing produced at North Carolina, Vascular Plant Systematics, and it was just full of information, including their suggestions for how you construct keys. So the nice thing about this is basically how every botanist in the world constructs keys. You decide what group you're going to do. You don't really do all this. You don't prepare a full description, but you at least put together enough information to where you know what characters are important in distinguishing one plant from another. You select key characters with what they call contrasting character states. Okay. And then they suggest you put together a comparison chart. And I think for a big group, you have to. This is a mandate. Strictly dichotomous keys. That means that a key only has two choices at each level. I personally think that sometimes it's fine to construct what's called a trichotomous key, because sometimes you've got one oddball plant and you might as well leave it out rather than have to manipulate the entire rest of the key to keep that plant, put that plant in the right place. But anyway, in the world of botany, keys are strictly dichotomous. There's two choices, two choices, two choices. Parallel construction. So the terminology in the key, if the point you're making is about the leaves, then both leads on the couplet have to address the same thing. Leaves alternate versus leaves opposite. What they like you to do is try to come up with characters that break groups into even chunks. So you don't pull them out one by one. It's really tedious to read through a key when every couplet in the key just pairs off one plant, one plant, one plant, and you're just kind of anxious to get to some group that is close to what you think the plant you're looking at might be. They like you to use at least two characters per lead. This becomes awkward because sometimes you've got one good character and then you have to kind of come up with something else that is supportive. You follow a format. We'll look at that. You always start the lead with the same word, like leaf broad versus leaf narrow. You always use the plant part first and then the descriptive phrase after that. And then they suggest that if you have the time and the plant has many different states, like a sedaceous plant, you might want to construct different keys that address different, like the flowering and the fruiting stages or something like that. So they show two examples of keys. This is called a bracketed key. And in this way, this is the simplest way to do a key. You line up put your plants in categories, you make your combinations, and you come up with the characters you're going to use, and you break the group into two parts based on your strongest character. And then you just start. You start with number one, shrub or vine versus herbs. And then you choose one of those, and they're going to choose two. So woody vine versus shrub, that's number two. Three, alternate versus opposite. Four, pinnately vein versus palmately vein. So you finish up everything under the first lead, and then you number the next one, and that becomes, so this number takes you to two, two takes you to three, three takes you to 4 to 5. But up here, this one took you to 6. So that is a simple way because you can just kind of sit down with material and write it out. A little bit more complicated graphically, but nicer as far as the user goes, is what's called a yoked or an indented key, where the characters include all of the plants that fit within them, so that shrub or woody vine, so everything that keys out as a shrub or woody vine is here. Herbs, everything that keys out as an herb is down here. And then you see the same couplets. This is the exact same keys. So this is the same as 1. That's 2. That's 3. That's 4. And then that's 5. And then back down to 6. So you could number these just as they numbered the others. Let me go back. Just as they numbered these. Okay, now, then they include suggestions for how you use keys. So choose the appropriate key for the place. And that's pretty logical, right? And the truth is, the smaller the region treated, the easier the key will be to use. It is easier to use Cluel's key to the panhandle here than it is to use the key to the whole state of Florida, because the key then includes, the Florida key includes all of the tropicals down in South Florida that aren't even present here. So when you key out something, you have to wade through all that. So if you have a regional key, that's just a lot easier. Suggestion: read both leads of the couplet before making a choice. You got to do that because you have to be able to kind of compare and make sure you're making the right choice. Otherwise, you just waste a lot of time. Of course, check the meanings of terms. Um, this one's kind of hard. You're out in the field, you've got one plant. Do not base your decisions on a single observation. Well, that's because keys are written in herbaria. And typically in the herbarium, they've got a whole folder of plants. So they are not basing it just on one specimen. They're looking at all of them. But but when you're identifying plants out in the field, you may only have one. Try both choices. That's sort of the same as looking at both, but you may find it's very exhausting. Here you're standing out and the bugs, you're sweating, you're in full sun, you're having to shelter the book to read the key because the sun's so bright. The bugs are driving you crazy. The wind is blowing and you're looking at these characters. So um, so it's not so easy to be so patient as to sit there and say, well, let's run down both sides of this, but that's what you often have to do. Um, and then read the description and compare the specimen with an illustration. Once again, out in the field, not so practical. So what they don't tell you about keys, they don't tell you that the keys are created in an herbarium. And that means that the people who, sorry, the cat wants to get out. Okay. The cat is very demanding. So they're created in an herbarium using a dissection microscope and using a stack of specimens. So that's very different from the life that the rest of us lead. I don't have a great dissection scope. I've got one, but I don't have one. And especially out in the field, I don't have one. I've got my hand lens and that's it. The more you know about a plant group, the easier key. So what they don't tell you is if you know anything about the plant group, it allows you to manipulate the key and just to say, those don't count. It's not that. So it helps you with making your decisions on the couplets if you know anything about the plant group. They don't tell you that since keys are requisite, it's necessary. It's a professional necessity for botanists to write a key because if you don't write it, no one else can if you're the expert, right? So you really have to write the key or you've not completed your study. But that means that sometimes there are no really good characters that instantly distinguish one plant from another. The distinguishing factor is kind of a combination of characters. And so, quite honestly, very often they will punt. You'll get down to some couplet in a key, and you just look at it and think, that doesn't make any sense. I'll never see that. And certainly I can't look inside the seed and see whether the embryo is attached in a certain way. And the seed's so small, I can't tell whether it's got hairs or not. In fact, it's the wrong time of the year. It doesn't even have seed. So some couplets work for the professional and are necessary to complete the project, but don't do you any good. Once again, that ties to this. The key may be useless if what you're looking at doesn't have the right parts. Distribution matters. Once again, the tighter the region that your key applies to, the better off you are as far as identification goes. And the other kind of weird thing is that especially when it comes to pubescence, you'll find that an author may be consistent in how they use terminology, but it may vary slightly from one author to the next. So you're going to have to take all that into account when you use keys. So let's go back to um, uh, Wonderland and Hanson, and we'll take another group. Let's take Polygula. I've put some attention to this because I've been running into them in the field and I wanted to find out more about them. So this is their key. Um, and here is the sort of the same key, but that's a yoked key, right? 1 and 1, 2, 2, 7, 7. Okay. If you go to the atlas, you'll find essentially the same key, but presented as a bracketed format: 1, 1, 2, 2, 3, 3, 4, 4. Okay. Now, that's great and it's useful, but there's a lot more to be learned from keys than just what the identity of the plant is. So, what can we learn? Well, we can learn what the author thought the most useful diagnostic characters are to identify plants in a group. We can learn how the author defines a species based on looking at the characters that show up in the key. We can learn whether the key is totally artificial. That is, it doesn't end up grouping plants that belong together. It's just identification, or natural. In the natural key, they fall out in groups that actually reflect their evolutionary relationships. Um, you can uncover shortcuts. If you read the whole key and analyze it for a minute, it makes it a lot easier to sit down with the plant and try to identify it. Um, Something absolute is when you use a key out in the field, then you know instantly how well the person who wrote that key understood the group. If you lived in Missouri, um, you would be using a flora by a guy named Starmark, and his keys are fabulous. And the reason they're fabulous is because Starmark really knew that flora personally. He grew and collected and studied those plants for decades, and he was um, relentless and incredibly energetic. So when you use the Starmark key, you know that it's going to work for you, not all keys. Hmm. And you also learn by using the key what what are things that might be most useful in field identification. So now what I do is different. Um, I take the key, if I really want to understand what the author's trying to tell me, and I load everything into a spreadsheet. So here I've created a spreadsheet from the Florida Atlas to their um, bracketed key of Polygula. So you can see, and I've color coded for you the leads, and I like color coding anyway. So here's perianth, orange, yellow to green, and the next lead in this couplet. So there's the couplet, 1A, 1B. The next lead's down here. The fun thing about this is once I put it in this format and I put down the key number, I also put down the key history so that this tells me that this is 2A and its history is that it's 1A, then 2A. And so the green brackets are under perianth orange. They're either going to be inflorescence is branched or the inflorescence is a solitary raceme. We'll look at this in a minute. So then falling out under that are these characters. So by doing this analysis, I can sort this and play with it. And it also just helps me remember it. Let's look at what, so here's Seneca. So perianth orange to yellow with a branched inflorescence. Many of you will know these plants. This is either going to be, we're going to call it, we're going to use the new name Seneca, Polygula or Seneca, Cymosa, and Seneca ramosa. So here's ramosa, the short one that occurs at the edge of wet spots. And here's cymosa, the taller one that occurs in slightly wetter spots. And you will find these plants close by each other, but typically in little colonies, not as much intermixed as adjacent. So then we move on in the key. So we had branched versus inflorescence solitary. And that leads us to two groupings. These are difficult. if you had never seen the flower, you might not be able to distinguish between these. So wings long acuminate versus wings cuspidate to mucronate. That means the two wings that stick out from the flower are stretched out in this case, and in these, they're more rounded, and there's just a little point, a little tail, or a little mucro, a little point on there. And I don't think those are great characters. And so this is to me a punt. They just, we're going to do this. And the reason it works is because these plants, let's look at the distribution. So I live here on St. George Island. I'm never going to see, until I end up visiting people in South Florida, I'm not going to see smalley. The plant that covers the state that I'm going to run into is Nana. It's the little flat one with the kind of greenish yellow flowers. Okay. So, I'm not, I can, in my world, I can just strike out smalley. So now I've got Nana, and the same is true down here. So the wings are cuspidate. Well, that might be hard to figure out. That leads to Lutea and Rugelii. Once again, I live here. Rugelii is peninsular, but it's not reported from the panhandle of Florida. So that helps me a lot. All of a sudden, that key feature here is not 4 plants, it's only 2, Nana and Lutea. Let's just look at the rest of the key. So here's Rugelii, by the way. And when you see this, then you know it's a very different plant. If you know Polygola lutea, then, and you see these pictures and these herbarium specimens, you know that's very distinct. Just a second. Okay, so here's the rest of the key in my spreadsheet. So we looked at the perianth was yellow. Here's perianth purple. There's 2 breaks under that. The inflorescence is branched or it's solitary. And the branched, all it does is tie in this little white version of Rugelii that's called Baldwini. And then all the rest of the Senegas, Polygolas are down here. Here's another break. 9A, leaves whorled versus 9B, leaves alternate. We'll just go through this quickly. So if you get down to all the leaves alternate, all of a sudden you have one of those single breaks. You have incarnata versus everybody else. And they break into racemes dense versus racemes sparse. And you might not know this, but there are a lot of polygolas that have flowers scattered along the peduncle, and there are others in which they are sort of compressed into a head. So here's the ones that are sparse. So let's get out of that. Let's look at the same key in FloraQuest. So in FloraQuest, what's fun about this app is it starts out in each family and in each genus with some description that helps you understand the plant. So here they're telling you about the distinctive flower structure of Polygola, Polygolaceae, that there are 3 fused petals into a tube, one of which is called the keel. The one that's most ornamented is the keel. And then there are 5, the calyx is 5-lobed of, they say 3, but I just think of it as 2. 2 of the 5 sepals are the large ones that are called wings. And they stick out. In fact, in the Polygola flower, typically the most visible part is these 2 sepals. And they stay throughout fruiting. So they are one of the reasons Polygola flowers look colorful for such a long period of time. Okay, so they've got this, and in your phone, or this is on my iPad, you can choose this species. And so in this whole family, the Asimia breaks out, and then you get to the genus Seneca. Okay, if you go to the Seneca, see, and you can see it here, you can see their sequence. That's in their world, it's the DKQQ Polygolaceae Seneca. And And you can touch this and it'll show you a description of the genus. Notice this filter part right here. So you can actually touch that, and then you can choose one of 4 regions in Florida. And if you choose to shut out the others, if you live in South Florida and you close down all the others, it will adjust everything in the list and in the key to only be the plants that are thought that are reported from South Florida. So that solves one of the problems of keys. It gives you the capacity to make this key regional. Now, let's say that here, choose this lead. If you choose this lead, you're going to go to this key, 1A. But you can touch that button and you can see everybody that's in there. So you can shortchange all of this key work by looking at the list of everybody. If you decided that 1A is your answer, here's everybody in 1A. And if you know any of these and you know the plant you're looking at is not that, then all you do is touch this button and it will take you to photographs of that plant. So there's a lot of advantages that have just come literally since February to our offing. So let's walk through their key. So now I took their key and I put it in a spreadsheet. So here's their choice 1A versus their choice 1B. So everybody that has basil leaves and orange, yellow, or greenish white flowers is in this bunch. That's the list we just saw, those 8 plants. Then they break it out versus whether the inflorescence is a single pom-pom or a mini branched corum. So all of a sudden now we see that Simosa and Ramosa are down here. Baldwini is right here. Nana is in this group, and Ludia is in this group. So it's kind of the same characters as we saw in the Wonderland key. They're just remixed because there are only so many characters. And quite honestly, botanists learn from each other and copy each other's keys and adulterate or alter people's keys. And so you're going to see the same characters, but you're going to see them from a different viewpoint. And that's why it's kind of fun to compare them. Let's look at the rest of his key really quickly. So if the leaves are not whorled at the base, are not, if the leaves are different, the stems and the flowers are pink, purple, white, or green, except for Boykinii, then you're going to end up here. And then if the leaves are whorled at the lower nodes, you're in these plants. And if there are no lower whorl leaves and they're all alternate, then it's the rest of the key. And that if you follow the key in FloraQuest, these are the choices. So they're numbered 1B, and within 1B, there's 9A and 9B. Okay, now let's just go. So here's the 9B choice. The leaves are alternate. And within that, there's 20A and 20B. And the reason they're 20s is because they've already used up the other numbers in the previous selection. So the leaves are linear versus leaves broader than linear. Okay. And within linear, you come to that beautiful incarnata, which is very distinct from all the others. And also, you end up with this funny thing, cetacea in here. They say that, but these leaves are really linear, by the way. In fact, they're almost bract-like. I got into Polygala because I ran into 2 populations of this plant, and I keyed it out, and it was either Mariana or Chapmania or perhaps Natalia. So now I have this, and I've looked at it in this key, and the difference is inflorescence bracts dropping from the axis promptly versus inflorescence bracts persistent. That's not such a great character from the field. It is a good character in the herbarium. But in the field, you don't know what promptly means. How promptly do they have to fall off to fit in this category versus that? So at that point, you have to do a bit more working. So I went to the Florida State University, and there's the herbarium cabinet that has all the polygolas in it. And they have, this is uh, Benjamin Ajayi, and they have a dissection scope, and you can look at the specimens, and then I can go and look at the other key. This is the key from the atlas, and I can see that it's not just it's not just deciduous. They also note that the inflorescence apex is rounded for Mariana, and the apex of the inflorescence tapers to a point for Chapmania and Natalia. So here is here are specimens of Mariana, and that's the inflorescence. Here are specimens of Chapmanii, and notice that there's this funny little thing. It's a curious little thing, but if you look at the flowers, then you see that they probably correspond to this. And also, they still seem to have some of those minute bracts. That's not definitive, but I've got a reasonable ID. So let's look at keys in a totally different way. Let's go to the daisies, to the composites for a minute, and talk about how the keys work in composites in the Asteraceae. Once again, if we go to FloraQuest, he's got a complete description of the family, and he gives you his terminologies, and it's elaborate, almost exhausting, in that suddenly the word radiant and the word radiate indicate different types of heads. And so at least he explains all that, because these words are going to work into the keys. Now, going back to another source, if you look in the flora of Florida, you will find a massive key to the composites. So this is so massive that it's 168 couplets, and there's an index key that takes you to 5 different keys. And then you start, this is exhausting work. I'm going to tell you that when you open a book and the keys are like several pages, you just take a deep sigh. So what I have done for my own understanding to get my head around the composites is I analyzed the entire key to the composites. I put everything in the spreadsheet where I can sort it and play with it. They don't tell you this, but key 1, everything in key 1 is in one subfamily. It's all the plants that have totally ligulate and perfect flowers. So they're all in the chicory, the lettuce group, Lactuca, and only one of them is not in there. So this first key, I wish they would put notes in there like I did. That if you get to know the family, you can skip all this and you can go straight to key 1. Okay. Here it is. So I've colored each tribe with a different color so I could kind of keep track of them. So everybody in the first key, except this genus, Anaportum, is in the chicory. Now, there are a lot of issues with keys that make them both functional and a little bit more difficult. And one is that a plant might show up in several places. So in this key to the genera of composites, the genus Gilardia shows up in 1, 2, 3, 4, 5 places. And remember, there are 5 keys. So it shows up twice in key 2. It shows up twice in key 3. It showed twice, 3 times Noscal and Soga, and it shows up 1 time in key 5. So Gilardia is, you'll run into it. It's such a unique plant. You just wish they would just show you a picture and say, this is Gilardia, and leave it out. Gaillardia is in the group with Helianthus, so it's in that tribe. So let's take a section of the Helianthe, because I can sort this now. And the tribe Helianthe is all yellow. So here's Gaillardia, Galensoga, and this is Helianthus. Helianthus shows up in two places. It shows up in key 2 and it shows up in key 5. Why is that? Because key 5 is for plants that don't have arrays, ray flowers. And if you know Helianthus radula, you know that many times there is no evident ray flower, or it's so reduced that it's hardly evident in radula. So of all the species of Helianthus, the one that is the exception means that Helianthus shows up in two different places in this huge key. So let's look at Helianthus. Here's how you, so, and the reason I'm doing this is because I want to get my head around what the authors think makes Helianthus a good genus, like what holds Helianthus together. So this is the history of all the leads that lead up to Helianthus in key 2. They're radiate. The rays are orange or yellow because that's a big division. The disc flowers are fertile. The basal leaves are opposite or whorled. The ray flowers fall off of the developing achene. So when the fruit is developing, the ray flower falls off. The receptacle, it has chaff. It's got palea. The filaries are more than 5. The achene is thick. It's not flat. The ray flowers are sterile. Some leaves are always bigger than 7 millimeters. There's never a time when a plant has every leaf is less than 7 millimeters. If it does, then it's in the allied genus Fabanthus, right? The pappus is of 2 awns, and they fall off, and the plants have leaves up the stem. So that is, and this one is exactly the same, except for the fact that the heads are discoid. So everything else is pretty much the same. And so by examining the key and reading it, all of a sudden I have a pretty decent description of the genus Helianthus. But when you read the description of it, here's the flora. When you read all that, a lot of that is kind of rote, like the leaves are opposite or alternate, simple pinnate, the flowers are in heads. Well, all daisies flowers are in heads. That's hardly worth saying. The ray flowers are zygomorphic. Well, pretty typically, yes. The petals are 5. Well, in the disc flowers, pretty typically they have 5 petal lobes. Basically, yeah. Stamens 5. Yeah, it's a composite. Epipetalous, yeah. So this description, which is a formal device, once again, doesn't serve you so well because the description is written according to a formula, and it's not a diagnosis, it's a description. So now I've taken Helianthus and put it in a spreadsheet as a way of analyzing it. And you can see that for the genus, the first split is basal leaves are well developed versus the basal leaves either are not well developed or not present, and the leaves go up the stem. So in these plants, these species here, the flower stalk either has very reduced or no leaves at all, whereas in these, the leaves are well developed all the way up the stalk. And this, by the way, goes all the way here. So just these few are like that, and all these rest have cauline leaves. And then our next choice is the disc flowers are reddish versus the disc flowers being yellow. So all of these plants have red colored disc flowers, okay? And all of these, the disc flowers are yellow. And then they start breaking them out. Now notice that some plants are going to come out twice. Let's see. Anyway, Angustifolius, disc flowers yellow. And now we break them out one at a time. We peel off Angustifolius, we peel off resinosus, We peel off Floridanus, tuberosus. That's painful. Okay, we finally get down here. And if the stem is glabrous versus the stem is hirsute, and that's when we end up with these two species and these two. Okay, so that's how that key is structured. But let's look at what the real truth is. Of these species native to Florida, Atrorubens is only known from Jackson County. Dericatus is only reported from 2 counties. Occidentalis from those 3. Microcephalus from these few counties, and resinosus from these. Heterophilus is all east of the Swanee River here, I mean, west of the Swanee River. And then hirsutus and stimulosus have this distribution. So if you live in South Florida, 1, 2, 3, 4, 5, 6, 7, 8, these species you're unlikely to encounter. So let's look at the ones that are more widely distributed. Angustifolius is statewide, and Radula is mostly statewide, all except, of course, this more tropical area down here. And then Debolus is represented by at least 3 different in the FloraQuest 4, 3 different subspecies. And you can see that one is up here, one is down here, and one is along this. This is Cucumerifolia, and then these 2. Agrestis shows up in the middle of the peninsula. Floridanus is lower coastal plain, and Carnosus is endemic to Florida. And then there are 4 non-natives: Annuus, Argophilus, Simulans, and Tuberosus. So that's the whole picture of Helianthus. Suddenly, if you live in South Florida and you've got this key, you can knock these out. If you're operating this key, that's the word we use, you operate the key, then there's only 2 that have basal leaves that you might encounter. One is Radula, and the other one is Carnosus. Okay. Of the ones that have cauline leaves, you're not going to encounter these. So it changes the formula for you. If you know this distribution, if you're studying the key, you can go and simplify the key, make it a lot easier to interpret. By looking at the key characteristics, the ones that were down here, then I can define Helianthus differently. And I can looking at the description in the key. So Florida marks the southeastern extent of the genus, and that becomes obvious when you look at Flora of North America. In North America, there are over 50 species, with ranges extending from North America to Mexico. Of those 50 and in Flora Florida says there are 70, but of those 50 plus species, 14 are reported native to Florida, and another 4 have been introduced. So now I can tell you that a nice way to discuss this, we have 14 native species and 4 introduced ones. These are the introduced ones. These 4, I'm sorry. These 4, Agrestis, Radula, Floridanus, and Simulans, are coastal plain plants, and Carnosus is our only endemic. Debolus is represented by 3 to 4 subspecies. So let's look at what constitutes Helianthus. There are annuals, 3 species are annuals, all the rest are perennials. They're erect to spreading herbs. Their basal leaves will be opposite. Some species are characterized by conspicuous basal leaves. The remaining 13 have cauline leaves. The heads are typically singly borne on peduncles, or just a few in an open corym. The involucres are green and somewhat leafy, except in Radula, the flowers are radiate The rays are yellow or orange. The ray flowers are sterile. The disc flowers are fertile. The pappus consists of two awns that are caducous. They fall off at maturity. The receptacle has pales. The palea are folded. The bracts, the chaff, each chaff wraps around and clasps an achene. The achenes are not usually flattened or winged. They are terete to tetragonal. So that's very different from the description. Let me go back. That's in the flora. This is the description in the flora because that's the way botanists describe a group formally. It's kind of formulaic. By reading through the key and understanding the key, all of a sudden you're able to construct a description that might allow you out in the field to look at a plant and say, you're going to be a helianthus. I don't know which one, but you're going to be a helianthus because you have these characters. And basically, I've taken the key and completely condensed it into one functional diagnosis. So I'm trying to convince you how much fun keys are. I know that probably is wearing you out, but don't just think of a key as something that you use to identify a plant. A key is really the secret information that the botanists don't get around to telling you in a more normal format. So what are the take-home lessons? Well, let's go back over what I've said. First off, keys are academic. They're based on herbarium specimens. They're really not for you, even though theoretically they're put in books for you, but they're not for everyday naturalists because they actually include characters that don't work for you in the field, typically or often. They're prescribed. They respond to a protocol. Keys are they have to do this and they have to do that, even though sometimes that's not logical. They are best when they're regional. The location makes all the difference in the world. If you've got a regional key, then it's going to be a much smaller key and a lot easier to operate. And not only that, they're able often to use clearer characteristics because you're not trying to include all the oddball things. A key is personal. It absolutely reflects the thought and experience of the individual or the team of people who put it together for a flora or a monograph. Keys are copied. Usually, a flora will tell you where the key came from, but it's like a cookbook. The best cookbooks are the ones where the author actually has tested and worked with and many times served or prepared everything in the cookbook. But there are a lot of cookbooks that are compilations and they just went out and they found somebody's that they trust, somebody's great recipes for broccoli and somebody else for eggplant and someone for lamb. And they kind of looked at them and decided which was best and put them in the cookbook. Well, those may be useful, but you can tell the difference because when you make a recipe that the author may have never made, it's unlikely to work as successfully as one when the author literally understands exactly how that recipe works. The same is true of keys. So where they got the key really depends on, the quality of the key depends on where they got it and who wrote it. Keys are hypothetical. Once you get to an answer, you cannot rely, you've got to go and verify that. You need to go look at some photos, some herbarium specimens, some identified material to confirm that you've reached the right conclusion. Keys are typically artificial. So just because you've worked through a key and you get to two plants or a group of plants, that doesn't mean they are a natural group. It's nice if they are, but it doesn't mean that they are. Keys are forgiving. Keys often allow for variable solutions. You saw that Gaillardia showed up several times. So you need to expect the fact that a plant will show up 2 or 3 times, trying to make allowance for the fact that you don't quite understand what the key's telling you, or the fact that the couplet doesn't work for that one plant. And then keys are summative. They give you a fabulous outline to the most diagnostic characters of plants. So if you're getting interested in a group, just sit down with the flora, review the key, make some notes about it, and you'll find that is the most valuable thing you can do to get your head around the entire group. So with some citations and sources, I think I will finish that. Let's go back to a prettier picture, something with some color to it, maybe. Let it sit. Oh, a lot of color. Yeah. Okay. So I'm going to stop sharing now and turn it back over to Rubs and Emily.
All right. Well, thank you so much, Jim. We've gone just a few minutes over time. It's fine. And we don't currently have any questions on the board. So again, just a reminder, if questions come to you later, feel free to email them to us at info@flawildflowers.com. .org, sorry, info@flawildflowers.org. And I really wish that at this point we could give you a virtual standing ovation because this entire series has been absolutely incredible. And we're just so thankful for your time and your expertise. And, we just hope everyone will stay tuned for more opportunities with Jim. As I mentioned, we'll be in the field with him in October, and maybe some other learning opportunities will come up as well. So um, I can't say thank you enough. I know we've received so much great feedback from this series, and we look forward to continuing to work with you.
Okay, I'm
Thanks everybody for joining us.
when possible, I'm delighted to answer questions. So if something is totally hopelessly confusing, be sure to write. And or if there's something that I left out that you think should be included, let me know, because all of this is documented in my website and I can go in and edit that. Thank you very much. Thanks for doing all this work. You guys, that's been a lot of effort on your part, and I appreciate it all.
Fantastic. Well, everyone have a great rest of your day, and we'll see you next month.
Okay.